mixed species richness on those islands. Get into this idea of the equilibrium theory of biogeography. And then third, and just sort of really briefly at the end, sort of compare and contrast sort of all these different types of dispersals that sort of appear towards the end of your second section. So for starters, if we're to think about species diversity and how it's distributed across the world, the first sort of pattern that really jumps out at us is that species richness is not evenly distributed. There are places in the world where the species richness is not very high. You go there, and if you're looking at plants, you might find only a few hundred species over like 10,000 square kilometers. You can go to other places, and sort of in that same 10,000 square kilometers, you might find 10 times as many species, or more, or even 100 times as many species. And there's sort of a general pattern to it. So in this map, basically the reds and the purples indicate areas of the highest known plant species richness in the world. And then sort of the olives and brownish colors indicate areas of very low species richness. And so the one thing that should jump out is that this is not evenly distributed, that different numbers can end up in regions that are very species rich and other regions that are very species poor. And it roughly tracks where you find the equator, which appear to be areas of relatively high species richness, and as you get towards the poles, you end up in areas of much lower species richness. And we don't have to just look at plants or continents. For the aquatic among you, if we were to look at marine diversity, we sort of see very similar patterns, although not in the ocean. So here we see sort of the same general trend as we go towards the equator. Tend to be more species in the equator, less as you get up towards the north and the south pole. Tends to be a little bit offset here in Southeast Asia. You get in Australia incredibly high diversity, also areas of very high diversity just off the coast of Africa. Things are a little weird over here in the New World, where our areas of really high diversity are not right at the equator, but still in the tropics, basically up in the Caribbean. But the general pattern, this idea that species evenness or species richness is not evenly distributed, holds not just for plants, not just for marine diversity, but for amphibians, for mammals, for reptiles, for a whole range of organisms. Look at an example involving ants. This is just another map, again with the red and orange colors indicating areas of very high diversity, the blue indicating areas of lower diversity. And it appears, just like with marine systems, just like with terrestrial plants, that there appears to be some sort of relationship between species richness and latitude. This is sort of a little bit of a wonky graph, but I'll walk you through it. So on the y-axis, our response variable is species richness. So it ranges from 0 on up to about 7,000. So each one of these would be a different species. And then the way you read this graph is right here in the middle, 0 degrees would represent the equator. And sort of by convention, as we go from the equator south, we go to negative 40 degrees latitude, or positive 40 degrees latitude to go towards the northern hemisphere. And in both cases, we see this general dropping off of species richness as we move from the equator out to the mid-latitudes, sort of represented here by 40 degrees. And then as we continue beyond 40 degrees, we see the diversity continues to decline all the way up to the Arctic. Things drop off a little bit faster here in the southern hemisphere, basically because our ants run out of land. So this latitudinal gradient is something that we'll come back to on Thursday. But for now, I'd like you to just sort of recognize that species richness is not evenly distributed, and because of this sort of pattern associated with latitude, it sort of demands a bit of an explanation. So the area of biology that deals with the geographic distribution of species richness is called biogeography. Sort of a nice mix of bio, life, and geography, sort of where things are. It's basically the sort of my shorthand for biogeography is the science of where everything is and how it got there. And because I'm a biologist, everything to me is referring to all the species. So biogeography basically addresses the geographic distribution of species richness. Does a little bit more than that, but we can use that as a starting point. And we can divide biogeography sort of into two major different groups. We can have ecological biogeography. And ecological biogeography is going to focus largely on more short-term interactions among organisms and between those organisms and the physical environment. So these can include things like predator-prey relationships, mutualisms, competition. It can include things like areas where organisms are able to maintain heat balance, or where evapotranspiration allows vegetation to persist, or particular species persist or not persist. So it's focusing on these more short-term interactions among organisms, between among organisms and among organisms in the physical environment. Historical biogeography focuses on more long-term evolutionary processes. And again, sort of thinking back to the very, very beginning of the course, there's sort of a common misconception that, when we talk about evolution, that sort of means long periods of time, and ecology means sort of very short periods of time. But as we saw with the examples with our cotton bollworms, evolution actually happens very rapidly from generation to generation. So I put the term here, long-term evolutionary processes here, specifically because I'm not interested in short-term evolutionary processes in the area of historical biogeography. It's these long-term processes, such as speciation, adaptive radiation, extinction, stuff that we'll get to a little bit on Thursday. So much of the things that happen over millions of years and tens of millions of years. So they both historical biogeography and ecological biogeography come into play in sort of this discipline of biogeography. In some years, we do offer a biogeography course in biology departments. That's something that I've been teaching for about 12 years now. And so I would just sort of encourage you at this point, don't think of biogeography as just sort of one little topic in ecology. We're basically glossing over many issues just in this one chapter. But there's all sorts of really interesting bits of biogeography that can easily expand out to 15 weeks or ideally a 2-semester sequence. And I get excited, but I'm going to try to contain myself and just do a little bit of biogeography. I know some of you have also signed up for an online tropical ecology course. This also has a significant biogeography component. It's probably about 3 or 4 weeks of biogeography in there. Some of which is labeled as such, some of which it's not. But again, I get excited about biogeography. So, sort of biogeography in the sort of grand sense. What I like to do is sort of move into that first learning objective and look at these ideas of alpha, beta, gamma richness, gamma diversity. A lot of times in the ecological literature, in books or in papers, people will use the terms diversity and richness interchangeably when talking about alpha and beta and gamma diversity. They're always talking about richness, but a lot of them are sloppy and will use the term diversity with that richness. Just something to be aware of. But if we're thinking about species richness and why some places like in the tropics are incredibly species rich, other places like the poles are species poor in the grand scheme of things. this sort of presupposes that we know how to go out and physically measure diversity in particular places. And so, by way of introduction, we'll look at alpha diversity, or the number of species occurring at a single homogeneous site. So it's going to be all the species from one particular location. In the Simitex lab, you were sampling on a wall, sampling species richness. And I wanted to sort of show you what the wall actually looks like. Almost cool. Can't quite get that graphically here. But essentially what researchers will do in order to generate these kinds of curves would be to sample sort of little 1 square meter areas on these coral walls in coral reef ecosystems. And it would generate curves that effectively would look like this for hard and soft corals and other sessile organisms. So alpha diversity is the number of species you find at a single homogeneous site. And there's sort of this problem you sort of encounter if you start thinking about it. You know, what's a site? Simitex just has sort of this rock wall as a site, but Wright State Woods, could we consider our campus woods a site? Or like, is it 2 sites because a road goes through it? You've got sort of one side and another side. Or if you're studying lichens, an individual tree might be a site. Ecologists, of course, they can't even agree on what a site is. A lot of times, when ecologists are talking about a site, they're talking about sort of the smallest unit that they're actually going out and physically measure. So, one ecologist might call the whole Wright State Woods a site. Another one might call 2 separate sites with a road running in between. Either way, what is important to focus on is not what they're defining as a particular site, but specifically when they're referring to alpha diversity, what is the unit of measure, and what kind of information about species richness are we getting from that particular site as defined by that researcher. I'm going to contrast alpha diversity with gamma diversity, or regional diversity. I'm not a big fan of the words alpha diversity and gamma diversity. They sound confusing, they sound pretentious. I think local diversity and regional diversity would work just as well. They wouldn't let me make up the terminology. So gamma diversity or regional diversity is just the number of species found throughout a particular region. And if we wanted to get really specific, we would call that the regional species pool. You think of the regional species pool as sort of a pool of species the same way you might think of a gene pool as a distribution of genes throughout lots of different organisms in a breeding population. So the concepts are analogous. So again, what's a region? If we're looking at the Wright State Woods, and we all agree the whole woods, one site, what would the region be? Okay, the Ohio, one possibility, eastern half of the United States.
So it depends on specifically what the study is looking at, depending on what type of interactions it's looking at, what type of tools it's looking at, small photo.
Okay. It could depend, okay? And I think all 3 of these answers are in fact correct. Again, just as we have sort of trouble sort of nailing down exactly what are we talking about when we talk about an individual site, we could run into the same problem when we're talking about a regional species pool. Right? What's the region? Is it just Ohio? Is it all the eastern United States? Speaking of which, where's the eastern half of the country end when you go west? Mississippi, but there's forests on the other side? Maybe we can include the forests on the other side, but maybe not the grasslands? Yeah? All right. I don't know. Mississippi River seems like an artificial barrier to me. But you're the ecologist, you're going to take responsibility for defining your regional species pool. All right. So we have a regional species pool, and then each of our local communities, local community 1, local community 2, local community 3, represents a subset of all the possible species we find in the regional species pool. About 20 years ago, when I was working as a postdoc, I had a project where I was looking at vegetation in Wisconsin forests, wildflowers. And we had 62 different sites that were sampled at 2 different time periods, 50 years apart. And we basically, from those 62 sites, from 1950 to 2000, we were able to sort of construct a regional species pool that sort of spanned that whole 50-year period. And that regional species pool basically was just every unique species that we encountered either in the 1950s or in the year 2000. So I think we had about 300 species in our regional species pool. And our regional species pool was just looking at vascular plants that grew in forests in northern Wisconsin. At each one of our sites, each one of our 62 sites, we had a subset of that regional species pool. Each site did not have 300 species, or even 200 species, or even 100 species. Typically had far fewer. And so we'd go out and we'd sample, much the same way you might sample that coral reef wall, the coral quadrats. And 20 square meters, we'd come up with about 25 species on average that declined through time. For various reasons, now about 20, but in the 2000s, it still continues to decline. But in this particular case, we had a regional species pool, which basically we constructed from all the information we had from the local species pool. And this is the way people typically do it. If you want to know gamma diversity, you need to have lots of alpha diversities, and to take all that information and sort of glom it together to get that regional species. So we have this, the city text sort of put forth this question of what determines how many species you would find at a single site, or in other words, what determines my alpha diversity for a single site. And they put forth sort of 2 different hypotheses, one of which is that alpha diversity is just proportional to the regional species pool. So if you had 300 in the regional species pool, you would basically just sort of have 25 randomly selected species at any given site at all 3 different sites. Sure, some might be 23, some might be 28. But local sites are just basically random samples of what you find in the regional species. Alternatively, alpha diversity would be limited by strong species interactions. So in other words, it's not just some random assemblage of species, but instead, that community is strongly structured by bottom-up forces and top-down forces that affect community structure, influenced by predation and mutualism and competition. And so these species interactions basically limit the number of species that you would find at a site level or at an institutional level. And if you were to graph it out, those 2 hypotheses basically show up as this blue line and this red line here on the bottom. Let's start with the black line. Start with the black line because it's almost not what it's talking about. The black line is basically just sort of a perfect slope of 1. So here you have 20 on the x-axis, 20 on the y, 40 on the x-axis, on the y. This is just sort of a way of orienting yourself to the graph. It has no intrinsic meaning associated with the data per se. So under hypothesis 1, this idea that each local community is effectively just a random sample of what you find in the regional species pool. If that was the case, that as you continue to sample over, or when your regional species richness got higher and higher, your local richness would just sort of increase linearly and proportionally. It wouldn't be quite on the black line because if it was right on the black line, it would mean that local richness was the exact same thing as regional richness. just can't pack all of your species into one little local site. It's always going to be less than and just sort of proportional to. So the slope of the proportional line is not as steep, but it continues to rise in a linear fashion. And under this model of alpha diversity, it suggests that species interactions generally aren't important in determining what particular species belong to the location. However, if you saw a situation where your local species richness sort of rose, but eventually plateaued and leveled off, rather than continuing to rise proportionally, this would suggest that the species would sort of fit within model 2, or the hypothesis 2, and the communities effectively become saturated. This is what you'd expect to happen if species interactions were very strong and ended up limiting what species would colonize a local site. So proportional and saturated, you can sort of think of as species in which interactions among species are unimportant versus interactions among species are very important in determining what appears in a local.
So the lines in the red line is basically hypothesis 2.
Hypothesis 1, hypothesis 2.
And then what's the black line?
The black line is, mathematically it's called a unity line. It's showing direct proportionality. So it has a slope of 1. And it doesn't have any biological meaning. It's just a way to help orient you to the graph. Yes.
So can you think of it as kind of like a control to compare it to?
The black line, or no, the black line is not even a control because a local site cannot contain the entire regional species pool. It might confuse you. It might not confuse other people. Yes, good for them. This is essentially a theoretical maximum. You can never be in any space above this line. So it's a bounded problem. Bounded. So what did semiotext say about the importance of species interactions in local communities? Question on it. Someone say something?
Oh, he's taking a nap.
I'm sorry, did I repeat it? What did semiotext say about whether or not local communities appear to sort of follow this hypothesis 1 of proportional diversity or hypothesis 2 of sort of saturated diversity?
It's always saturated.
Say again?
Saturated.
Saturated. No, not saturated. There's a whole figure. Over lots and lots of sites, it's very hard, but most of the data actually suggests most of what we see is proportional diversity in particular places. Not strictly proportional, but it suggests that the role of species interactions is quite weak in helping influence local species. Go back and read that part of semiotext. You might get that question on the final exam. All right, we've looked at alpha diversity or local diversity. We've looked at gamma diversity or regional diversity. And I hopped right over beta diversity. I skipped it entirely. We're going to get to beta diversity now, because beta diversity is sort of the weirdest part of this whole alpha, beta, and gamma diversity. It's a little bit different. So beta diversity It's basically a measure of the change in species within a community as you move from one local community to the next local community to the next local community. And so what this figure shows is first of all, 1, 2, 3, 4 separate local communities. Each different geometric shape represents a different species. So here we are over in local community 1. We've got an alpha diversity of 1, 2, 3. Alpha diversity 3, right? Now we have 3 species. We have red triangle species, we have green square species, and we have blue star species. We move on over to the next local community over, and we've got, it's remarkably similar. We've got red triangle species, green square species, blue star species. 100% overlap. No change. Those two communities are basically identical. Go down here, holy crap. We've got red triangle, green square, blue star. Same community over here. Go over to the 4th community, red triangle. Oh, we've got a new species. We've got, not sure what color that is, yellow pentagon, and we've got yellow circle. All right. So basically what we're seeing here is a lot of species similarities. We move from one community to the next. The way we'd say it is has very low beta diversity, or alternatively, we'd say it has low spatial turnover. Because as we go from one community spatially to the next one, spatially to the next one, we tend to see the same species over and over again. We contrast this with local communities exhibiting high beta diversity, high spatial turnover. So here in community 1, we've got yellow pentagram, yellow circle, and blue star. We go over to the next community. Only one species still the same. We've got blue star. Now we've got red triangle and green square. Down here, kind of different. We've got green square again. We've got yellow pentagram, yellow circle. Over here, pentagram, triangle, star. Okay, so each one of these communities is actually pretty different from one another in terms of the species composition, unlike you see over here in sort of low beta diversity. Same example. So low beta diversity, relatively high beta diversity. So let's look at just some real data on a comparison of alpha and beta diversity in bird communities. If we were to go out and do bird surveys at individual sites throughout North and South America, red represents areas of very, very high alpha diversity or local diversity. And we see places like in the Atlantic forest of Brazil, in the Amazon, up through northern South America, a little bit in Central America, we see very, very high diversity. As we move away from the equator towards the poles, that diversity tends to drop off. It's kind of low by the time we're into the mid-latitudes like we are here in Ohio, where the equivalent mid-latitudes down around Argentina, Chile. Alpha diversity has that sort of classic pattern that we see like with plants, like marine systems, and with ants, sort of that really strong latitude gradient. However, if we were to look at beta diversity, and the change in diversity as we went from one local community to the next local community to the next local community spatially, we get a very, very different sort of pattern. In general, we still have a stronger influence in the tropics. It's lower in the mid-latitudes. But there are places in the temperate zone, in California, even all the way up in Alaska, where it gets kind of high again. Get very, very high beta diversity of these birds. So, what's going on here? Yeah?
There's some fairly elevated areas from what I'm seeing. The mountains near Chile.
So what So why would the bird communities change just because there's mountains there?
Because high up in the mountains, they can get there a lot easier than a lot of other animals.
But these are just birds, they're all birds.
You got the mountaintops kind of act as islands and they fix the corners of different mountains, one area to the other.
Okay, so the mountaintops can sort of act as islands in the sky that sort of can keep animals sort of lot from getting from one place to another. It's a possibility.
It's just like as you go from like Amazon to the Andes, like the Andes are much like colder and drier, so you're not gonna find like toucans and parrots and stuff that you can find
Largely correct. You do find toucans and parrots higher up in the cloud forest, but you find different species of toucans. You find the mountain toucan instead of the channel-billed toucan. You find different species of parakeets in the cloud forest than you do in the lowland rainforest. So what you're largely right. As you move up in elevation, the vegetation changes, the resources for those birds change, and as a result, you get different species of birds that are able to persist in those areas. So even though you're moving over very short distances in terms of kilometers, you get very, very big changes in the kinds of species you're encountering. So that sort of helps explain why in the Andes we have this really high beta diversity in through the mountains of Central America. We also have several other mountain ranges where you get those sort of really strong changes in species composition as you move from one location up in elevation to another. And you see that trend sort of persist in here through the west, in the Sierra Nevadas, and best place to get in here. I don't know the geography of Labrador or Alaska well enough to explain what's going on there, but presumably those are elevational related as well. Alaska's probably not. You get very different species in the lowland tundra than you do at
It's like you got one or the Alaska glaciers.
and different birds in those islands. Yes.
It also be human factors, because some like the East Coast is a little more mellow.
It could be, but also the other thing you get on those eastern areas, actually you're probably right, it's probably largely anthropogenic influences, but you also have that coastal influence as well. So you have changes associated with marine birds versus terrestrial birds. So alpha and beta diversity are different ways of looking at diversity. I was thinking of asking you guys a question. If you guys want, so that you get into pairs or small groups, I'm going to place you in charge of managing a 5,000 acre nature reserve as forests and grasslands and wetlands. You're evaluating two different management options created by brilliant scientists. One management option focuses on maximizing alpha diversity within that area, and the other plan would maximize beta diversity. Both would cost the same, both would have sort of the same level of support from the public, funds are available. If you had to look at the trade-off between alpha and beta diversity, which would you choose? Would you choose to sort of maximize alpha diversity or maximize beta diversity?
So how was the diversity in the different
beta diversity, right? What do you guys choose? I think beta.
What?
I think it's beta diversity, yes.
No.
Because alpha seems like it's only a smaller location or area versus beta is like the regionals. And 5,000 acres sounds like a lot.
That is a lot. So you'd rather have
And I think for beta, alpha, it's more of like a specific locations. But the reserve it consists of forest, grassland, wetland. It's a lot diverse. So I think for regional, it sounds a lot more makes sense or peaceful. Does that sound right?
it sounds reasonable. We'll discuss it a bit.
Oh, okay. So it's not like a right or wrong. It was like
It's
something.
not wrong. It's reasoning through and justifying your explanation.
Oh,
and
okay.
taking responsibility for what you're arguing.
Okay. Cool. What do you choose? You don't know? You think it's alpha? Oh, why do you think it's alpha?
Well, one, I don't think it's two, because we're talking about such a small area. 5,000 acres is really not.
Oh, really?
I mean, it's bigger than Wright State. When you look at, like, compared to
The regionals?
yeah, it's not that.
Not that big? Okay.
So,
I guess, yeah. But they have like many of the area, like forest, grasslands, wetlands, whereas like alpha is like, I feel like it's more specific one location.
well, okay. I guess ours is a very homogeneous site because it's got the forest grasslands.
Unless you control individually,
Yeah.
because I guess the turnover between the environment would be, I would love.